Hi, I'm Carol. I will be talking about a little bit about epigenetics and diabetes type 2, also known as TD2. So maybe I'll be referring as TD2 from now on. Maybe I'll be a little bit repetitive now, but as my colleague stated before, epigenetics is basically the gene regulation patterns, right? So if we remember the last content of this class, and that we talked about the chromatin structure, the DNA packaging. Now, if we take a closer look to the histones, the proteins that plays this packaging role, and they have some tails that can interact with different compounds, and promote some events as methylation and acetylation, which can promote the relaxation or the packaging of the DNA, turning on or off a gene. And this regulation of turning on or off a gene can be regulated by other genes, for example, tumor suppressors that prevent the cell to divide itself a lot. Also can be regulated by environmental factors, which is the case of diabetes type 2. But just to state a little bit of difference between the two, diabetes type 1 is genetically defined mostly, and diabetes type 2 is mostly lifestyle related, but also has some genetic compound, because if you have a family history, you may be more susceptible to the environmental triggers that can actually lead to this disease. Yeah, that's it. So what happens in diabetes type 2, right? Mainly the insulin resistance, so the body cells become less sensitive to insulin, which is really important to regulate the glucose levels in the bloodstream. So beta cell dysfunction as well. These beta cells are present in the pancreas, which produce the insulin, and in TD2 it may become less efficient or even damaged over time. It also can lead to inflammation, so kind of low, chronic inflammation that can contribute even more to this insulin resistance and the beta cell dysfunction as well. So what genes are involved in it? I've seen a lot of articles and they mention different genes, but I choose to talk only about one, the most reported one, which is the GLUT4, which stands for glucose transporter 4. It is a transmembrane protein that stays in the membrane and basically uptakes the glucose from the environment and follow its metabolic processes. I will spare you from the biochemistry and all these biochemical details, please. But in the TD2, this gene that codes for the GLUT4 is methylated, which means that the DNA is super packaged. So what that means that the transcription factors cannot access the DNA. And if you cannot access the DNA, you cannot produce the RNA messenger, you cannot produce the RNA transporter, and you cannot produce the protein and follow with the central dogma of biology, which you will discuss in the post. Because not always are coding for proteins, but that's it. This happens, and it is really prevalent in this disease. There's a lot of other genes that plays roles in the development of this disease, but I will just keep on this one. Yeah, so that's it. I'm curious to know what you guys think. If you have questions, I will be happy to respond. I love talking about epigenetics, so it would be nice to discuss with you guys. And that's it.